So I'm going to talk about biology of neuroendocrine differentiation. So the in benign human prostate, we can observe two kinds of epithelial cells under H&E sections. So these are luminal cells and these are basal cells. And there is a third type of epithelial cells, which is a minor component, which we call neuroendocrine cells. They cannot be visualized on routine H&E sections, but if you do immunohistochemistry using a marker of neuroendocrine differentiation, you will see them. You can also detect them by electron microscopy. So once you have prostate adenocarcinoma, what you have is you have malignant proliferation of luminal cells and you have loss of basal cells. But every single case of prostate adenocarcinoma also has these scattered rare neuroendocrine cells. And in some cases, the neuroendocrine cells can be quite abundant. They form these clusters. So I often actually have, receive phone calls from my clinical colleagues. They say that, well, I have a patient and the pathologist called the tumor prostatic adenocarcinoma with neuroendocrine differentiation. What exactly does it mean, and what do I do with it? I usually tell them, I said, I have to look at the slides, because if a pathologist look at the adenocarcinoma, he suspects there are neuroendocrine cells in them. He does immunohistochemistry. He sees more than usual number of neuroendocrine cells. It probably does not mean anything, at least for now. But if it's a small cell neuroendocrine carcinoma, then the disease, it's a different disease. Well, we'll talk about small cell neuroendocrine carcinoma later. So you can also see this by electron microscopy. You can see the dense core neurosecretory granules. So this kind of tumor cell heterogeneity is really remarkable, because I'm a pathologist. Just up until a few years ago, I was looking at all kinds of tumors, and we know that there's tumor cell heterogeneity at the molecular level. But for a pathologist to see this kind of consistent heterogeneity, I have to say prostate cancer is the only one. Because every single case of prostate cancer, we see luminal type cancer cells and we see neuroendocrine cells. So why are these neuroendocrine cells important? Number one, unlike the non-neuroendocrine tumor cells, these are the chromogranin negative, these are luminal type cancer cells. They express very high levels of PSA, but these neuroendocrine cells in tumor, they do not express PSA. Therefore, at the late stage of the disease, after multiple rounds of different treatment, very often the patient tumors have abundant neuroendocrine cells. And because these neuroendocrine cells do not produce PSA, very often the tumor burden and PSA level do not correlate very well. And as some previous speakers have commented on this fact, that at the late stage, PSA level is not very useful in determining disease burden. The reason these neuroendocrine cells don't have express PSA is because they do not produce and they do not express androgen receptor. You can see here the non-neuroendocrine tumor cells express very high levels of nuclear androgen receptor, and the neuroendocrine cells are negative for androgen receptor. This is something else that I think may be very, very important, because so far most of the treatment that we do, we give to these patients, we assume that the tumor cells are homogeneous. So if we target androgen receptor is the most common target, but we assume every single tumor cell has androgen receptor. And now we realize that this AR targeted therapy is not curative, and lots of people are looking at ways of combination therapy to cure cancer. But I think one important area of combination therapy may be to co-target these luminal type tumor cells, using traditional approach such as AR inhibition, and as well as neuroendocrine cells. But so far, we don't know how to target these neuroendocrine cells. because they don't express androgen receptor, they are resistant to the to hormonal therapy and targeting androgen receptor. So you can see that in hormone-naive tumors, tumors that have not been treated with hormonal therapy, you only have scattered neuroendocrine cells. But in general, once you have castration-resistant tumor, there is abundant neuroendocrine cells. So what's the neuroendocrine cells in normal prostate and adenocarcinoma? The short answer is no. But a number of studies over the years have shown that these neuroendocrine cells have secretory functions. So they produce biogenic amines, neuropeptides, cytokines, and etc. And the adjacent non-neuroendocrine luminal cells, tumor cells, express receptors for some of the neuroendocrine cell products. Therefore, you can imagine a scenario where when you have an untreated tumor, you have mostly luminal type cancer cells and rare neuroendocrine cells. And once you give patient hormonal therapy, these luminal type cancer cells will die and you enrich for the neuroendocrine cells. And eventually, these neuroendocrine cells, by secreting their products, may be able to establish a local paracrine network to support these tumor cells to grow. So this is one hypothesis, which is very difficult to prove. A very important characteristic of these neuroendocrine cells is that they are quiescent. Dr. Isaacs earlier mentioned the fact that Ki67 may be very useful in predicting prognosis. I actually quite agree. I think the most important prognostic tool is Gleason grading, and Ki67 may be second, in my opinion. Except that we pathologists, we are kind of lazy, because I want to look at the slide for 5 seconds, make the diagnosis. I don't want to spend half an hour to count Ki67. So that is one problem. You can see that in these non-neuroendocrine cancer cells, you have, this is dead. I don't have the pointer anymore. You see Ki67 activity, but if you have neuroendocrine cells, they are always quiescent. They don't express Ki67. Oh, okay. Yeah, very good. Importantly, if the patient received hormonal therapy or the other AR-targeted therapies, a number of them can actually develop a tumor that is pure neuroendocrine cells, which we call prostatic small cell neuroendocrine carcinoma. But these neuroendocrine cells in this small cell carcinoma are different from the neuroendocrine cells we see in adenocarcinoma, because these cells are highly proliferative and very aggressive. So through a series of studies, we have come up with these hypotheses for the molecular mechanisms of by which the small cell carcinoma develops in after hormonal therapy. So what we have observed is that these neuroendocrine cells, they secrete IL-8. They also express IL-8 receptor CXCR2. And this autocrine action activates a p53 pathway and keeps these neuroendocrine cells quiescent normally. And during the course of hormonal therapy, p53 mutation happens, and which knocks out this growth inhibitor pathway, and these neuroendocrine cells become highly proliferative, and then you have a small cell neuroendocrine carcinoma. So I just want to show you some examples. p53 mutation is rare in untreated adenocarcinoma. Most of them are totally negative like that. But if you stain for p53 expression in small cell neuroendocrine carcinoma, vast majority of them give you a staining pattern like this. So about 75% of small cell neuroendocrine carcinoma have p53 mutation. And here is a tumor that is partially adenocarcinoma and partially small cell neuroendocrine carcinoma. You can see p53 mutated expression is only happens in the small cell carcinoma area. So in addition to p53, some other studies have shown that Aurora A kinase, MYC, RB, REST transcription complex, and pectin, they are all potentially involved in the pathogenesis of small cell neuroendocrine carcinoma. So in terms of the mechanism, there are two possibilities. My friend Colin Collins likes to believe in trans-differentiation model. He believes that during the course of hormonal therapy, the adenocarcinoma cells gradually transform into neuroendocrine cells. That's why you have small cell carcinoma. But I would argue that because they have distinct genetic lesions, p53 RB mutation, clonal selection or clonal expansion is more likely the mechanism. So I'm going to use the final several minutes to describe to you what we have seen with the samples from the West Coast Dream Team. So we have 300 men that we are going to enroll, and we are going to look at the metastatic biopsies, which gives us unprecedented pathological material to look at metastatic tumors. Because I don't believe any pathologist has had the chance to look at more than a few metastatic tumors up to now. So right now, I'm up to 180, patient number 183. And in these metastatic tumors, we see a spectrum of histology. So some tumors have this classic adenocarcinoma histology, and others have the classic small cell neuroendocrine carcinoma. To my surprise, I see a very large proportion of these metastatic tumors that have this kind of morphology that I didn't know how to diagnose. They don't meet the criteria for the diagnosis of small cell neuroendocrine carcinoma, but I really cannot diagnose them as adenocarcinoma. So finally, we decided to give them a distinct category. Initially, I wanted to call it atypical neuroendocrine carcinoma because they really look like the pulmonary carcinoid tumor. But I got Mark Rubin and Brian Robinson to come to UCLA to look at our cases. Mark insists that I cannot have the word neuroendocrine in them because he said, you have not proven they are neuroendocrine cells. So finally, our West Coast Dream Team leader, Eric Small, came up with this term, intermediate atypical carcinoma. And here is the morphological features of these three different tumors. I'm not going to go into detail because most of you are not pathologists. You wouldn't be interested in them. But anyway, here is the three representative pictures of these three type tumors: adenocarcinoma, small cell neuroendocrine carcinoma, and this intermediate atypical carcinoma, as named by Eric Small. So why is it important to distinguish this particular type? Because before I had this idea of putting them into a different category, I would say that because they don't meet the diagnostic criteria for small cell carcinoma, by default, they would be diagnosed as adenocarcinoma. But if you look at the Kaplan-Meier curve for patient survival, it is pretty striking. In these patients that I diagnosed as adenocarcinoma, by the way, when I had these slides, the slides is all I have. I didn't have any other history. I didn't even know the biopsy site for many of them. Some of them have liver or bone, so I know, but many of them I don't even know the biopsy site. So I just make diagnosis purely based on histological criteria. If my diagnosis was adenocarcinoma, by 22 months, 80% of these patients are still living. Remember, these patients have failed everything. By the time we did biopsy, they have really failed everything we have. They still live for a long time. If my diagnosis was small cell carcinoma, the median survival is only 6.6 months. If my diagnosis is this IAC, the prognosis tracks small cell carcinoma very, very closely. So it is very important to recognize that even though they are not morphologically small cell neuroendocrine carcinoma, their prognosis is similar to small cell carcinoma. Now I'm going to talk about some of the genomic stuff that our collaborators at UC Santa Cruz has come up with. And so every time when I talk about genomics, I get nervous because unlike Scott Tomlins, who is well versed in both surgical pathology and genomics, I am an old-fashioned pathologist, so I really don't know how to deal with genomics. But so I picked a few slides that are relatively easy to understand. So the Santa Cruz people tell me that when they did the unsupervised analysis, the small cell carcinoma that I have diagnosed cluster very, very well. But adenocarcinoma and IAC, they seem to be together. However, these people are really, really smart. So they have come up with a 50 gene signature, and they can now very confidently separate IAC from adenocarcinoma. And I was told to just focus on these numbers. So with their gene signature, they can separate small cell versus anything else, small cell versus IAC, small cell versus adeno, IAC versus adeno, IAC versus everything else. So they can, with this 50 gene signature, they can do it with great confidence. And they actually mentioned that as few as 5 regulators often accurate enough. So I'm going to stop here. I want to acknowledge the people who have helped me. These are people in my lab, and there are many collaborators that I've had at UCLA who have helped me tremendously over the years. And for this Stand Up to Cancer project, Eric Small is a PI, Owen Whitty is a co-PI, Rob Reiter and Chuck Ryan, they are major leaders of the team, and Matthew Reddick, Joy Thomas, and Larry Chu. Joy Thomas and Larry Chu are 2 pathologists who helped me, who confirmed my diagnosis. So because originally I didn't know if I was correct, so I gave them my diagnostic criteria. Using my criteria, they can diagnose them very, very accurately. And I will stop here. Thank you.
AR, what in the intermediate ones? How are they positive or negative?
We haven't done AR on every single case, but the few cases that we do, they are all positive. So,
and why isn't the word, which I used to think when I was growing up, anaplasia, I thought that was the good word for when you couldn't really say anything more about a carcinoma. In other words, it had no glands. So why are these not just anaplastic cancers?
So they are not anaplastic to a pathologist. To pathologists, anaplastic tumors are tumors with widely different tumor cells, very angulated, weird shapes, atypical mitosis and everything. So this particular tumor type does not have that. Cells look very homogeneous, look very low grade, even though their biological behavior is very aggressive. So I thought that, I read Anna Aprasic's paper. I thought maybe they were talking about these kind of tumors. But the anaplasia really does not apply here, because when pathologists say anaplastic, there is a specific morphological criterion that is wide differentiate, very wild variation in tumor size and shape, and just looks very highly malignant to the eyes of pathologists, and this doesn't.
So I've heard the West Coast and East Coast dream teams saying that the East Coast one was generally ABI and ENZA pre-treated, untreated, and the West Coast one was post-treatment. But I'm not sure if that's actually true, because if you look at the supplement of the Cell paper, which is the East Coast, actually like 51% of patients have received either abiraterone or enzalutamide, perhaps not both. So it's actually a misnomer that the East Coast dream team is only untreated CRPC, and the West Coast it was about 80%. So it's 80% versus 50%. So the first question is, have you looked at any of the pathology from the East Coast team and vice versa? Is it possible that you're both looking at the same thing and calling it different names?
So that is a great question. The differences lie in the fact that our protocols are different. So for West Coast Dream Team, we do several biopsies. 1 or 2 cores go to formalin, so we have paraffin fixed tissue, we have nice H&Es, like how I usually look at patient specimens. West Coast Dream Team, they are more focused on sequencing. So everything is done fresh and frozen. So they cut a frozen section slides. These kind of subtle morphological differences cannot be discovered on H&E slides. So I look at a few their slides, it's impossible.